bogs down, and some don't go in, they spill off for a while as acetone, these fruity smelling ketones. So, what does this mean? Well, the pH then of the body fluids drops, enzymes are less efficient, and the body's response is to carry out respiratory alkalosis, a little huffing and puffing. You're trying to blow off some of these acids. You get rid of CO2, you can get rid of some carbonic acid. So this results in respiratory alkalosis. We haven't talked about this, but it just means a little hyperventilation. You're blowing off excess CO2. That does remove some of the acid in the body. So this is a problem for people with diabetes mellitus. It's a problem for people that are fasting in general. And if you were to look at the natural history of the human race, we would see that the survivors that brought us here have on and off gone through this, haven't they? I mean, so this is a mechanism that we have that we can survive. You know, it's definitely survivable, but it does cause some specific challenges to the system. So let's think about lipid synthesis now, which is an interesting situation. It turns out that if you ingest excess carbohydrates, your body will store those excess calories as fat, okay? And so, we're going to talk here about glucose being transformed into triglyceride lipids. And this is something that we know about. I mean, when we, in our society, we can get the supersized sodas, right, with all this sugar in there. And if we drink these supersized sodas regularly, what happens? We don't excrete the excess calories, we store the excess sugar calories as fats, which is the most efficient way to actually store them. So how does this happen? Let's look at our chart. We said that lipids can be digested and the partial products shunt into glycolysis, and as the acetyl group they enter into Krebs. Well, the reverse can be true as well. If we have excess glucose, we can pull out glycerol. And if we have excess calories, we already said that if we have lots of ATP, it starts to negatively feed back on these pathways, right? And then we're going to pull out acetyl-CoA in this direction and build fatty acids. So glucose can make glycerol, glucose can make fatty acids. Joining fatty acids to glycerol gives us fats.
So what are you pulling out of the acetyl-CoA? What are you naming it there?
This is the reverse of beta oxidation.
Right.
This is what we call fatty acid synthesis from the acetyl groups. We're just joining 2 plus 2 plus 2 plus 2 plus 2 plus 2. We get up to about 16 or 18, we've got a fatty acid. It's just the reverse of beta oxidation. Fatty acid synthesis. Okay. Now, in this way, we have excess glucose, excess ATP results in lipid synthesis. All right. So let's now bring in proteins into the picture. With proteins, why do we need protein in our diet? We need protein in our diet because we need certain essential amino acids that we can't synthesize. We use those to build our own proteins. So with amino acids from proteins, these can also be used to make some energy. When you get into the starvation that the Donner Party experienced, they start digesting their muscle tissue, using protein to make amino acids, and to get some ATP from that product. So we're going to see that amino acids from proteins can serve as a source of ATP. Well, let's see how this works. First of all, let's differentiate the two major classes of amino acids. We have essential amino acids are the amino acids that human cells cannot manufacture. We have to ingest preformed essential amino acids. Humans require in our diets because we can't make them from scratch. E. coli, common bacterium in our gut, has the enzymes to make these essential amino acids. It's enzymatically smarter than we are. So we have to get these from the grocery store and provide in our diet. So how do we make amino acids? The non-essential amino acids are manufactured in the following way. Humans have the amino acids to manufacture the non-essential amino acids. And we can take a, let's say, amino acid number 3 plus, let's say, a keto acid number 6. Where did amino acid number 3 come from? Well, this came from our diet. We had excess amino acid 3 in our diet. Where did the keto acid come from? It came from either the Krebs cycle, or perhaps pyruvic acid, or perhaps somewhere in glycolysis. We have these keto acids we can pull out. They're intermediates. And watch what happens. We're going to now get keto acid number 3 plus amino acid number 6. What happened? We took the amine group, the NH2, from this, and we transferred it here. Just this molecule, literally, the keto acid 6, cannibalized the amine group from amino acid 3. That's all it did. It just stole it. So that's one thing that can happen. And then we can have
The amine group itself?
just the amine group.
Just,
So if we were to draw this, if we were to draw this, all you're doing is just taking the NH2 and transferring it over. Very common transfer type of reaction. Then, how do we get energy from amino acids? Well, if we take an amino acid, we can carry out a process called oxidative deamination. So what does deaminate mean? Remove an amino group. Decarboxylate. Remove a carboxylic acid group, CO2. So now we've got an amino acid over on the left and a keto acid on the right, plus ammonia. What do we do with this keto acid? This is the reason for the deamination reaction. The keto acid, depending on its structure, could go into glycolysis. It could go into the Krebs cycle. And so now if we provide ingredients for Krebs cycle or the glycolysis, we have a way of getting ATP from amino acids, which came from proteins, right? We digest the protein by hydrolysis, we get amino acids. The amino acid, deaminated, we can make ATP.
Okay, when you say it could go into glycolysis or it could go into Krebs, it has to go into glycolysis first. It can't go straight in, can it? Or
It can go straight
does
into
it go to
Krebs.
the acetyl
I'll give you, let me
the
give you an,
pre-Krebs?
let me give you an example. It could go right into Krebs.
Oh,
It could go into Krebs. There are several amino acids which, if you remove their amino group, they can either go into glycolysis, alanine can go to pyruvic acid, or they